Okay, so chapter 13, it's about viruses, viroids, and prions. So viruses are infectious agents that are considered in microbiology because they are very small to be seen by the naked eye. And although they are not microorganisms themselves, just the fact that they actually are pathogens, they live intracellularly, they are considered then in microbiology. Now, viruses can form films like this one in here, and with these films, they can transmit infections to humans or animals. Now, we're going to see in this chapter differences between viruses and bacteria. We're going to describe what are the characteristics of viruses, how we classify them, how we name them, and how we can grow them and study them in the lab. And we're going to see how viruses multiply, and we're going to look at the steps of viral replication. So here it's a table showing you differences between viruses and bacteria. Bacteria can or cannot be an intracellular parasite. We have few of them like chlamydia that are intracellular parasites, rickettsias as well, Ehrlichias, but the rest of the bacteria are not intracellular parasites. An intracellular parasite requires cells to replicate in them, but bacteria can or cannot need them. Now, viruses are always intracellular parasites. They cannot replicate outside cells. And when it comes to different structures, bacteria has a plasma membrane always, while viruses can or cannot. If a virus doesn't have a plasma membrane, it is called a naked virus. And now, in terms of replication, bacteria replicates most of them by binary fission, while viruses, they don't replicate by binary fission. Nucleic acid presence of both DNA and RNA. Bacteria has both DNA and RNA, while viruses can have one or the other, but never both at the same time. So then, based on these, we can classify viruses as RNA viruses or DNA viruses. And then for generation of ATP, some bacteria, it's able to generate ATP, some others they don't. And then when it comes to viruses, they cannot replicate, they cannot generate ATP. They actually need the cellular machinery of the host, like ribosomes, mitochondria, etc., to obtain energy. Ribosomes are present in all bacterial cells. They are capable of making proteins, while viruses, they don't have ribosomes. And sensitivity to drugs. So bacteria are susceptible to the action of drugs that we call antibiotics or antimicrobials, while the viruses, they are susceptible to the action of drugs like interferons and antiviral drugs. So here is general characteristics of viruses. Again, they are obligate intracellular parasites. They do contain DNA or RNA, so that's a major classification of viruses. And the structure of this nucleic acid can be double-stranded or single-stranded. So we have, for instance, single-stranded RNA viruses, which are the majority of them. And then we have double-stranded DNA, which is also another virus that is very abundant. And then we have double-stranded RNA viruses and single-stranded DNA viruses. And this nucleic acid can be linear, circular, or segmented. For instance, we have influenza viruses that are RNA viruses that has segments. We have like 7 segments made out of RNA. And then again, viruses, they don't have ribosomes and they don't have organelles like mitochondria. And they can be protected. Well, actually, the nucleic acid is always protected by proteins that we know as capsid. So we have a core for the viruses that it is called a nucleocapsid. In terms of viral symmetry, so we have viruses that are helical or rod shaped. And then we have some viruses that are called icosahedral. Which will be typically spherical with 12 facets or more. And then the viruses that are helical have protomeres. That surrounds the nucleic acid. While the icosahedral or spherical have capsomeres. Surrounding the nucleic acid. So those are the units. And these capsomeres or these protomeres are polypeptides. If it's a capsid, it's going to be a single type of polypeptide. And if it's a icosahedral, it's going to be different types of polypeptides. And then in icosahedral, you form a complex of DNA plus the capsomeres that is called the nucleocapsid. Now, attaching to this nucleocapsid or into the envelope, you have spikes. And these spikes can be glycoproteins that attaches into receptors in the host cell that helps them to be absorbed by the host cells and then create an intracellular infection. And then most viruses infect a specific types of cells in one host when the host is multicellular. So we have some viruses. Actually, viruses can infect all kinds of species. Okay? But we have some viruses that infects bacteria. And this bacteria has in their cell wall some receptors that are used by the viruses to attach into these cells. And then we have animal cells with receptors that are specific for the species in many cases. And also within species, we have a specific receptor that infects certain tissues. For instance, we have viruses that infect only the liver cells, causing hepatitis, but they do not cause infections anywhere else in the body. So that's the specificity. So the host range is determined by these specific host receptors, and cellular factors that are found within these cells that makes the viruses to thrive within these specific tissues. So it's more than the receptors, it's also what the cells have inside that makes them susceptible to infection by particular viruses. In terms of viral sizes, we have some viruses that are very small, others that are intermediate in size, and some that are very big. So in this figure, you can see different types of cells. Like in this case, these will be eukaryotic cells. This will be an erythrocyte. So an erythrocyte has a diameter of almost 10 nanometers. And then, almost 10 micrometers. And then in here, we have E. coli, which is a bacteria, it's a prokaryote, that measures 3 micrometers in width by 10 in length. So this is a gigantic cell, a red blood cell, in comparison to this E. coli bacteria. And now, one of the largest viruses that they exist, that looks like a rod or a sugar cane candy, it will be an Ebola virus with a length that almost, it is the length of this bacteria. So it's almost 1,000 nanometers or 10 micrometers. So this is a very long virus. And then we have this virus that measures 800 nanometers by 10 nanometers in diameter, that is called bacteriophage M13, that actually can infect bacteria like E. coli, that it is very long, although it's very thin. And then we have complex viruses like vaccinia virus, that is also a large virus. So this vaccinia virus measures 300 by 200 by 100 nanometers in diameter. And then we have here a bacteria. This is chlamydia, the elementary body, the infectious form of chlamydia, that measures 300 nanometers in diameter. So it's basically this vaccinia virus, it is the same size as this. And then we have other viruses like bacterial viruses MS2, that it is very small. It measures 24 nanometers in diameter. One of the smallest viruses as well will be rhinoviruses or poliovirus, that measures only 30 nanometers in diameter. And then we have T4 bacteriophage, which is a complex virus. It measures in length 225 nanometers in length. So these bacteriophages can infect bacteria, that's why it's called bacteriophages. Others infect animal cells like rhinoviruses, polioviruses. We have adenoviruses that also infects animal cells, or of course vaccinia and Ebola viruses. Others like Viroids infect plants, and these are not viruses, but they are made of only RNA sequences, and they have a diameter, these Viroids, of 300 in length by 10 nanometers. So it's like almost 1/3 of these bacteriophage M13. And then prions, which are proteinaceous infectious agents, they are not viruses. They are in length 200 nanometers by 20 nanometers in width. Now in here we have rabies virus, which is part of the Rhabdoviridae family. This rabies virus infects animal cells. It looks like a bullet shape. It measures 170 nanometers in length times 70 nanometers in width. And then here we have a plant virus, tobacco mosaic virus, that measures 250 nanometers in length times 18 nanometers in width. So there is no uniformity in general in length or in width in the viruses and in host. Some can infect plant cells, some can infect bacteria, some can infect animal cells. Now, the complete viral structure, which includes nucleic acid, includes capsid, and includes, in some of them, envelope with the spikes, it is considered a virion. A virion is a mature form, infectious form of viruses, and when it comes to the ones that don't have an envelope, will be called naked viruses, and includes the nucleic acid with the capsomeres and the spikes. Now, where the viruses that has an envelope acquire their envelope? Well, they acquire the envelope after they infect these cells, animal cells, and then when they shed, when they rupture these animal cells as they are exiting the cell, they acquire this coat, this envelope. So only animal cells will be enveloped, the ones that acquire the envelope. So bacteriophages, they don't have an envelope, they will be always naked. Now, depending then on the presence or absence of the envelope is whether the viruses will be susceptible to the action of some solvents like ether or alcohol or detergents. So the ones that have this envelope, they will be susceptible to the action of a solvent because this envelope is made out of lipids, and these lipids can be destroyed with these solvents like ether, alcohol, or detergents. But the ones that only have the nucleocapsid, but no envelope, the naked viruses, they are resistant to the action of this. So that's another classification, whether they are ether resistant or not. So we have here an example of a naked virus. So this is a capsid. These will be the capsomeres or protomeres, these structures that makes this capsid. And if you remove part of it, you can see that internally you have the nucleic acid. An example will be this virus that is called mastadenovirus. And these protrusions that you see, this is a transmission electron micrograph, are the spikes that this virus uses to attach into the host cells. Here we have a helical envelope virus, like influenza virus. This will be the nucleocapsid internally, and this will be the envelope. The envelope has these spikes that you can see here in this transmission electron micrograph that the virus uses to attach to the surface of the upper respiratory tract. Here we have a helical naked virus. So this will be the capsid with all of these capsomeres or protomeres protecting the nucleic acid, like, for instance, this Ebola virus, which is a helical virus. This is a transmission electron micrograph. This is one of the longest viruses. And these Ebola viruses infect the walls of our blood vessels, causing hemorrhage. So it causes a disease that is called hemorrhagic fever. Now we have, in this case, couple of images, in this case, scanning electron micrograph versus transmission electron micrograph. So this is a scanning electron micrograph of mass adenovirus. So these are the capsomeres. So this scanning electron micrograph, it gives you a three-dimensional structure versus a two-dimensional structure or flat image of a transmission electron micrograph. So this is a herpes simplex virus. This is an envelope virus. So this is the nucleocapsid, these are the capsomeres, and this is the envelope. This masked adenovirus, you've seen it before in a transmission electron micrograph, which is this. But see how this transmission electron micrograph, although it's very nice, this image, it doesn't show this three-dimensional structure of this. We have RNA animal viruses like rhabdovirus. In this case, we have this bullet-shaped virus that actually has members that causes rabies. And then we have here a filovirus, that it is a complex virus. It has these spikes in here. So this is the nucleic acid, this is the capsid, and then this is the envelope. It's an envelope virus. This is T-even bacteriophage, a complex virus. It's a bacteriophage, only infects bacteria. It has a head, which is this. It has a sheath, which is this part, and it has a base plate with tail fibers, which are these. And in the middle of this base plate, it has a pin that it uses to inject, as if it was a needle, its DNA into the host cells. So if you remove a part of this capsid head, you can see, this capsid in the head, you can see internally the DNA. So this is a transmission electron micrograph showing you the surface of this host cell, which is going to be a bacteria with the cell wall in here and three viruses here, three T-even bacteriophages, or phages for short, attaching into the cell wall. When the one in the middle, it already injected its DNA into the bacteriophage. That's why you see this empty space. While this one on the left side, this green color that you see here internally, that's the DNA still in the head. And this one on the right side, it partially has injected its DNA into the host cell. That's why you see this empty dark space in here, but part of the DNA, which is stained in green color, in here. And the DNA passes from the head into the sheath and then into this pin plate, pin into the base plate, and then into the host. Okay? So that's how this infection occurs in the bacteria. And then here we have a complex virus, envelope virus. This is orthopox virus. So here the nucleic acid, and then this is the surface of the virus. Now, viruses are classified into families. Families share homology in its nucleic acid. So family names end in the suffix viridae, and within these viruses families, we have genus names that end in the ending virus. And then within this genus, you have species. And species are viruses that shares the same genetic information, so they come from the same family, and they share the same niche. So they live in the same area. So we use actually common names for the species, and then within this species, you can have subspecies that infects in this niche, in the case for the human body, specific areas of the body. And in this case, the subspecies can be named with numbers. So here we have an example for members of the herpes viridae family. So the genus for this family that we are considering now is herpes virus. And within herpes virus, we have subspecies. So we have a subspecies that the niche will be the human body, so we call it human, and then herpes virus for the genus, and then we have a subspecies or subspecies of viruses that can be subclassified as type 1, 2, 3, etc. And in the case of abbreviation, for these viruses, we can use actually capital letters for both the genus and also for the family and for the species. And then you can use the numbers for the subspecies. And we have here these examples: HHV1, HHV2, HHV3. So that means that it's human herpes virus type 1, human herpes virus type 2, human herpes virus type 3. Okay? And now we have another example here for the Retroviridae family. So this is for a DNA type of virus, an example. Now for RNA type, we also have the same taxonomy. So we have Retroviridae, which is the family name, and then the genus name will be lentivirus. And within this lentivirus, we have some subspecies and species. So will be human immunodeficiency virus, that will be the species, and the subspecies will be type 1 and type 2. So abbreviation will be human immunodeficiency virus type 1, human immunodeficiency virus type 2, or HIV-1 and HIV-2. Any questions about taxonomy? Okay, so growing viruses in the lab is laborious, and then you always require cells. And the cells has to be specific for that species of virus. So if you're growing a bacteriophage, you have to provide bacterial cells. If you're growing animal cells, you have to provide animal cells of a specific type that has the receptors for that particular virus. So when you grow bacterial cells in the lab and you infect them with bacteriophages, these bacteriophages will grow internally in the bacteria, will make copies, and then it will burst these cells. And this bursting is going to create in the confluent lawn on the surface of the agar gaps that we know as plaques. And you can use these plaques to quantify the viral numbers for what we call plaque forming units, or PFUs. Now, if you're growing animal cells, animal viruses, you have to provide, again, animal cells, and you can use different types of cells, including cells coming from embryonated eggs, or cells coming from immortal cell lines or primary cell lines. Immortal cell lines grow indefinitely. They are derived from cancer cells. And when it comes to primary cells, they are mortal, so they have a finite number of replication. And another name for these cells that are non-primary cells, that are derived from cancer cells, are called continuous cell lines. So this is an example of what happens when you grow bacterial cells in the lab. So this is a top view of a Petri plate, and all these beige colors, as you see here, that it doesn't have gaps, are uninfected cells. But then these spaces are called plaques because there used to be cells there, but the cells get ruptured by the viruses as they were multiplying inside the cells, and they get released from the cells, causing cell death. So they used to be, again, live cells, but they died, and they produce these plaques. And these plaques can be quantified, and then based on the number of the ones that you have and the use of a particular formula, you can calculate how many viral particles you initially use to infect these bacterial cells. And you would do just similarly as you did before for the spread plate technique. You would do a serial dilution, and then again you would use your formula to calculate the PFUs, or plaque forming units. When it comes to using cell lines for animal cells, you will have cells that were separated with the use of some enzymes. And then these cells, you put them in a container where you have culture medium that it gives the nutrients for these cells to continue to grow. These will have amino acids, growth factors, and then you will have some antibiotics to prevent contamination of these cell lines and damaging to the cells with these bacteria. So depending on what type of cells would you have, if it's primary cells, you can have the growth of these cells in a single layer, or you can have growth of the cells in multiple layers, in which you have a continuous cell line or transformed cells derived from cancer, in which these cells have inhibition of cell-to-cell contact. Normal cells, they don't produce tumors. They're supposed to grow, and when they contact other cells, they're supposed to stop growing. But then tumor cells are invasive, and they don't obey that rule of cell-cell contact inhibition. So then they grow on top of each other, and then they can be used to grow viruses. So independently, whether you use primary or transformed cells, you can infect them with viruses, and then you can see what is the effect of these viruses when they're growing in these cells. And this effect is called cytopathic effect. Why is it called cytopathic effect? Well, because you can produce cell damage, and that it is like if it was a disease. Pathos means disease, cyto for cells. So these cytopathic effects are characteristics of that particular viruses, and then can be used for diagnosis or for research purposes. So here we have examples of embryonated cells where you can use different sites to inject viruses and to see these cytopathic effects. So here we have an egg that is embryonated. So this is the embryo. This is the chorion allantoic membrane. So you can use this site for inoculation. This is the other part. So this is next to this, this is the amniotic cavity, not the chorion allantoic membrane, which is this. So this is the amniotic cavity surrounding the embryo. So you can inject viruses into this amniotic cavity, or you can inject it in this membrane, or you can inject it in the yolk sac or in the allantoid, and then you can have different effects. Now, this is one way actually to grow viruses and then get some particles of the viruses to produce vaccines. Some vaccines are made in embryonic eggs, and this is one of the reasons why whenever you are filling a questionnaire and you're going to receive some vaccinations, they ask you if you're allergic to eggs, because some of these can be derived from eggs, and then it can produce an allergic reaction. So then when it comes to cytopathic effects, they are a specific damage to the cells that can be easily identified. Some viruses can produce clumps in the nucleus, others in the cytoplasm, or they can make the cells to die, the animal cells, or they can make the cells to fuse to one another, in which you call a syncytium when these cells clump, and you can identify this. So then a virus can be identified through the cytopathic effects or specific damage to the cells, but also can be identified by measuring the levels of antibodies or titers in a patient. And some can be identified by the use of antiviruses against these antibodies against these viruses, and you can use them in neutralization tests, viral hemagglutination tests, and Western blot. Others can be identified by amplifying a piece of nucleic acid, whether it's RNA or DNA, with PCR or polymerase chain reaction. And this is a very rapid test that can give you multiple copies of the virus's nucleic acid in a short period of time. So here's an example of a cytopathic effect. So here you have an animal cell. This is the nucleus. This is the cytoplasm with the extensions. And then after this cell is infected with virus and a neighboring cell, which is this other one, the cell dies. It retracts. Okay, so this is an example of cytopathic effect. Any questions so far? Okay. Now, viruses have a cycle for growing inside the cells. And the purpose of growing, of course, inside the cells is to multiply, to make copies. And just as when it comes to bacterial growth curve, we can make also a similar curve for measuring the amount of viruses that are produced and knowing the steps of viral multiplication. So depending on the virus type is whether the viruses can have two cycles, a lytic cycle and a lysogenic cycle. And then this viral multiplication will be actually in bacterial cells. So a virus has then two types of cycles, lytic or lysogenic. In lytic cycle, the viruses infects the cells, multiply inside the cells, and it ruptures the cells. Lysis is rupturing of the cells. In this case, the viruses, they do not integrate into the DNA of the bacteria. It only multiplies and ruptures the bacteria. In the lysogenic cycle, viruses enters into the cells. It can make a few copies, and then as the cells, the bacterial cells, the host cells are dividing, this virus can infect other cells But at the same time, the viruses can integrate its own DNA into the bacterial cell DNA, producing a hybrid. And that hybrid is going to be a provirus. There is an advantage of this. When a virus produces a provirus, no matter if it's animal cells, in animal cells or bacterial cells, the virus can survive for prolonged period of time within these host cells. When it comes to lytic cycle, the viruses multiply and can rupture the host cells, but if it doesn't find any other host cells to infect, the viruses can die. And there is a purpose for doing this. When nutrients are high, and when you have many host cells, because the nutrients are high, then it is advantageous for the viruses to multiply and then rupture the host cells and then find new cells to infect. So it can increase the level of infection. Now, when nutrients are low, the host cells, they don't multiply as much. And it doesn't pose an advantage for the viruses to multiply as much, because if it multiplies much and it ruptures the cells, then it won't be able to encounter new cells and to amplify the infection. So in that case, it opts for basically multiply at low levels and live within the cells for as long as possible, until the host cells now can find nutrients, multiply as much, and then the virus can multiply as much. So then you have to think about it in terms as if you are a virus, whether you have a lot of nutrients, multiply as much, amplify your infection, or maybe do not have as much nutrients, but being able to survive in a low level. So then, in a lysogenic cycle, you can form a provirus or a prophage in which the DNA of the virus incorporates with the host DNA, and also you can have in lysogenic cycle something that is called phage conversion, in which sometimes you can have part of the DNA of the host cell integrated into the virus, and when it goes and infects other cells, you can bring part of this DNA from the host cell, and in that case, the new host cell, if it doesn't have something else that it had before, now it's going to get it from this virus. And you will have something that is called a specialized transduction in that case. So here are the steps of this lytic cycle. First thing that it happens, you have to adhere the virus into the host cell, and this is known as attachment. And again, you have to have a receptor that the virus uses to attach. It's like a ligand for a hormone or for a growth factor. So you have receptors in the host cell. In this case, it's going to be the cell wall that binds to a spike or a tail fiber from the bacteriophage in this case. And then after that, you will have the next step, which is going to be penetration. In penetration, you have the actual crossing of the DNA material from the virus into the cytoplasm of the host cell. And in the case of bacteriophages, you create a hole in the cell wall with an enzyme that is called phage lysozyme. So the lysozyme then can create a pore within this cell wall, and then through this pore, the DNA penetrates into the cell. And then something that happens during this process is actually like that sheet, that middle part of the bacteriophage contracts, and then you insert the pin, and then the DNA goes through the internal part of the pin, just like if it was a needle, like if it was injecting, and then the DNA goes internally into the cytoplasm. Once inside the cytoplasm, the DNA of the phage will direct the cellular machinery to make more copies of DNA and as well proteins that will make the nucleocapsid, which is going to be protecting the DNA later on, and also to make proteins to make the base plate, the tail fibers, the pin, the sheet. So it's like an assembly line now internally in the cytoplasm. So you will make the nucleic acid, you will make proteins that are going to make the parts of the virus, and this process is known as biosynthesis, making the tiny parts of the virus. And then those tiny parts, they need to be assembled. So you put the head, you put the DNA inside the head, you put underneath the head the sheet, and then you put the base plate, and you put the tail fibers and the pin. And once you finish that process, you will have what is called maturation. And then once the virus is completely assembled, then you release it. And in order to release this virus, you have to have also a lysozyme enzyme to create a pore within the cell wall. So that's the lytic cycle. So then you can create a one-step growth curve of viruses in which you can count the number of virions in the y-axis and the number of days or time in the x-axis. So you will have on time 0 a specific number of virions, and then you'll have a period that is called eclipse period, in which this number of virions disappear, and then you have the number of virions increasing in this one-step growth curve until you reach a peak, and then decreasing. So that period from increase to decrease is called the acute infection. Now, if you see in this one-step growth curve, during the eclipse period, you have disappearance of the viral particles. They cannot be detected in the fluid or in your culture. Why is this? Well, because the virions first were found extracellularly, but then once they infect the cells, you cannot detect them in the fluid that surrounds the cells. It's not that they're not there in the culture, they're in the culture, but they're not in the fluid that is outside the cells. If you were to grab the cells, rupture the cells, in that case, you're supposed to get the entire number of virions back. But then, after this, the viruses start multiplying, making millions of copies. They reach a peak, and if you don't provide new numbers of cells, then just as for the plateau phase of your bacterial growth curve, then you will have equal numbers of deaths and equal number of new cells being created, in this case viral particles. And then since you don't have more new numbers of cells, then you will have a death phase. So it's exponential growth, but also exponential death, and you reach a plateau. So then this is the diagram showing you what happens of every stage of this infection in the lytic cycle. So here is a host cell. This is the DNA of the bacteria. This is the cell wall. This is the plasma membrane. And here we have attachment of this bacteriophage, a T-even bacteriophage. So this is the head, sheet, base plate, tail fibers. With the tail fibers, if you magnify it, it adheres or attaches into receptors in the cell wall. And then in the next step, which is going to be penetration, this sheet contracts. That makes this pin plate, pin in the base plate, which is this, to penetrate the cell wall, in which you also have here a lysozyme that helps to create a hole in the cell wall. And this DNA that it was found inside the head, now it passes through the sheet and then into the middle of this tail core, and you can see the DNA is injected into this part, which is the cytoplasm. Once inside the cytoplasm, this DNA is going to direct the synthesis of nucleic acids, tail fibers, sheet, base plate, etc. And this process is known as biosynthesis. The next step is going to assemble the different parts. So you have a head, you have to put the DNA into the head, forming a nucleocapsid. Then you have the tail, the sheet, and the tail fibers assembled. You have a complete virion. And then once you have complete virions, they can exit. That's the last step of this growth cycle. Any questions? So that's then the growth cycle. And that's lytic cycle. Now for lysogenic. So this is what we saw before, lytic cycle. Now for lysogenic, it is same steps. You have attachment and then penetration, release of the DNA into the cytoplasm of the host cell. And here we have DNA of the host cell in purple color, while in pink color you have the DNA of the virus, which is this. And during lysogenic cycle, this DNA of the virus is going to integrate into the DNA of the host cell, creating a hybrid through recombination. And now this DNA is going to be called prophage. Now, this virus or this prophage is going to divide every time that the bacteria divides by binary fission. And when the virus encounters that the host cells have nutrients available, it can excise out of this prophage, forming only DNA virus, and the DNA from the bacteria will be separated from there, and then it can undergo the lytic cycle, multiplying the virus, forming new virions, and that will lead to lysis of the cells, and then the spreading of the infection. So lytic cycle is favored when you have abundant nutrients, lysogenic cycle when your nutrients are low. Now for transduction by bacteriophage. So a phage will infect a donor cell. And then the phage and DNA proteins are made. The DNA of the phage and proteins are made. And the bacterial chromosome will be broken into pieces. When you have this DNA broken into pieces from your donor cell, part of this DNA can be integrated into these empty heads during the maturation process. And now, when these viruses lyses the cells and then encounter new cells, the recipient cell, which is going to be different from the donor cell, is going to be infected by this bacteriophage that carries DNA from the donor cell. And now, this recipient cell, through recombination, is going to grab a piece from this donor cell, and now it's going to have a different property. That different property can be maybe being able to utilize a nutrient, or maybe having resistance to antibiotics, disinfectants. In this case, it's going to be called a specialized transduction. But in here, then you will have basically a piece of bacterial chromosome carried by this bacteriophage into a new cell, which is going to be the recipient cell. So specialized transduction can be then given when this prophage, which is basically a copy of a bacteriophage integrated into the bacterial DNA. In this case, this bacterial DNA has a gene that is called galactose gene that allows this cell to utilize a monomer of a carbohydrate that is called galactose, coming from lactose, sugar. And then this donor cell, which is called galactose positive donor cell, when it is allowing the multiplication of this prophage, and the prophage undergo the lytic cycle, then when this prophage now carries a piece of this galactose gene in its DNA, now infects a galactose negative recipient cell, and this galactose negative cell can become galactose positive cell after this bacteriophage injects its DNA, carrying this copy from the galactose positive cell. And now this galactose negative cell, when it integrates this DNA from the bacteriophage, now it's going to become galactose positive recombinant cell. So now this lysogenic cell can use galactose, and previously wasn't able to use galactose. So that's a specialized transduction, and that's how sometimes these bacteria can gain an advantage. Now for multiplication of animal viruses. So you have attachment, just like as for the bacteriophages, you have to have physical contact between a receptor from the animal cell and a receptor or spike from the animal virus. After this attachment happens, the viral particle is going to penetrate the cells, and that penetration is going to happen by endocytosis, in which the virus can be encoded within a vesicle, or it can leave its envelope within the host cell envelope. In this case, it's going to be called fusion. And the nucleocapsid now is going to be found inside the cytoplasm of the animal cell. So after this, you have to release the viral nucleic acid and enzymes, if it has them. And then with this nucleic acid, so basically the capsid will have to be disassembled in uncoating. And then you can start with these enzymes, either from the host or the viral enzymes that the viruses can carry. You can start making copies of the virus nucleic acid, and also you can make proteins to start the biosynthesis process and then maturation. Now, in maturation is just assembling of the nucleic acid and the capsid proteins. And after maturation, you will release the viral particles by budding, in the case of the envelope viruses, because they will have to carry a plasma membrane within them, or by rupturing. If it's a naked virus, it doesn't need to carry any envelope, so it will rupture the cells. Any questions? So basically it's the same steps with the exception of uncoating. In bacteriophages, you don't need to have any uncoating because there is no capsid that penetrates into the cytoplasm. Only you inject the DNA. So there are no RNA bacteriophages. And then in the case of animal cells or animal cell viruses, you have uncoating. So here we have examples of viral cell entry and exit. So here we have a retrovirus that can be encoded with this vesicle. This is called receptor-mediated endocytosis. Or you can have fusion. In this case, this will be the envelope of a herpes virus. This herpes virus is going to leave the coat here within the plasma membrane of the host cell. So here we have the exit. After this lentivirus has made copies of its DNA or of its nucleic acid, and then puts a coat of proteins around the nucleic acid, this virus is going to get near to this plasma membrane, and then it's going to bud, so it's going to be surrounded by this envelope, and then it's going to be protected with this envelope. So this is the process of giving part of the envelope. Another example of viral entry. So here we have toga virus attaching into this plasma membrane of the host cell. So this dark line is the plasma membrane. All of these will be the viruses. The viruses has attachment spikes that attaches into receptors, so these will be the receptors, and then they will be endocytosed. It's brought as a vesicle. And then in this case, this is a herpes virus. We saw this before, so this is the nucleic acid, this is the capsid, this is the envelope, and this is the plasma membrane. And then it fuses its envelope, which is this, into the plasma membrane. So the envelope will be left in the plasma membrane. And then the nucleocapsid internalizes into the cytoplasm. So all of this is the cytoplasm, and then the virus will encode. Any questions? Okay, we will stop here and we will continue with this replication.